Welcome to The Explainer. Today, we are stepping into the high-stakes, mostly invisible world of clinical microbiology. I want you to think about the last time you gave a medical sample. A drop of blood, a cotton swab, a little plastic vial. We tend to view these as just routine tests, but really, we should be looking at them as incredibly delicate, precious cargo embarking on a perilous hero's journey. Because honestly, every single step of that journey, from the moment it leaves a patient's body to the final report landing on a doctor's desk, determines whether someone gets the right life-saving treatment or goes down a very dangerous, potentially fatal path.
So, picture this. You feel absolutely terrible, you drag yourself to the doctor, and you just want antibiotics, like yesterday. But they stop you, and they firmly insist on taking a swab or a sample first. Why the holdup when you're clearly sick and just want to feel better? It's incredibly frustrating, right? Well, that simple, totally relatable medical mystery is actually the key that unlocks the entire rigorous logic behind our microscopic cargo's journey through the healthcare system.
To get to the bottom of this, here's our roadmap for today. We'll start inside clinical microbiology, go over the rules of collection, talk about transporting delicate cargo, identify high-priority specimens, and finally, read the results. Let's dive in.
Part 1. Inside Clinical Microbiology.
Now, the primary mission of the lab is pretty straightforward on paper. Detect, identify, and figure out the characteristics of microorganisms so your doctor can give you an accurate diagnosis. They use this strict 10-step workflow to manage the whole life cycle of a test, right from the initial request down to the final report. It looks super organized, but here is the terrifying part about it. Every single one of these 10 stages can introduce catastrophic errors. Seriously, a tiny mistake at step 3 means the result at step 10 is basically just a complete lie.
Which brings us to Part 2, the rules of collection.
Remember that question we opened with about why you can't just take your antibiotics right away? Well, collection rules are insanely precise. If you take those meds early, you might literally kill the evidence. It can reduce or eliminate viable microorganisms, causing what labs call false negative or reduced yield cultures. The lab is going to look desperately for the criminal, the bacteria making you sick, but your early antibiotics have already wiped the crime scene clean of clues. Now, there is a realistic caveat here. Doctors are practical. They won't refuse to collect a specimen if emergency treatment has already started to save someone's life. They just have to document that treatment really carefully and adapt.
Okay, let's play out a real medical exam scenario for a second. Would you reject an unlabeled specimen? Just think about it. You know there is a sick, anxious patient waiting on these results. Do you try to guess whose sample it is based on what time it arrived or what it looks like?
Well, the hard truth is you absolutely must reject it. Medical professionals cannot just invent an identity. It feels brutal to throw away a hard-won sample from a sick patient, but they have to follow strict rejection policies and demand a new one. Why? Because processing an anonymous sample could lead to a fatal mismatch. You could end up handing one patient a devastating incorrect diagnosis or pumping incredibly toxic medication into someone who doesn't even need it.
Moving right along to Part 3, transporting delicate cargo.
Once a properly collected specimen actually enters transit, the danger isn't over. It faces massive threats, like rapid bacterial overgrowth or outside contamination. To defend against this, labs use a literal arsenal of specialized transport media. You don't just toss a swab into a dry plastic tube and hope for the best. You use chemically engineered environments. Stewart's medium is great for certain bacterial swabs. Amy's is your standard go-to for many common swabs. And Carrie Blair? That is the heavy-duty choice for enteric, meaning intestinal pathogens, like what you'd find in stool samples. They are perfectly matching the chemical armor to the exact medical need to keep that sample frozen in time.
And here's another awesome scenario from the source text. Why use Carrie Blair specifically for those stomach bugs? Because the stool sample is literally swarming with millions of fiercely competing bacteria. Carrie Blair is brilliantly formulated to keep the target enteric pathogens alive during the ride to the lab, while strictly putting the brakes on their multiplication. If those bacteria were allowed to just multiply freely during the car ride, the harmless background bacteria would completely overgrow, and the lab would never be able to spot the actual culprit making the patient sick.
This brings us to Part 4, high-priority specimens.
Look, not all medical cargo is treated equally. Some specimens are total VIPs that demand extreme speed. Take the standard midstream clean catch urine sample. Seems totally routine, right? But scientifically speaking, urine is basically an all-you-can-eat buffet for bacteria. It is a fantastic growth medium. Because of that, any delay in processing it can completely ruin the diagnosis. If that little plastic cup sits on a counter at room temperature for just a bit too long, a tiny, harmless amount of bacteria will rapidly feast, multiply into a massive colony, and totally trick the doctor into diagnosing a severe urinary tract infection that doesn't actually exist.
Then there are blood cultures. These are utterly critical for detecting septicemia, which are infections right in the bloodstream. For this, aseptic, 100% sterile collection is strictly required, non-negotiable. Why? Because a simple accidental brush of the patient's skin flora, those everyday harmless bugs just hanging out on your arm, can slip into the vial. To the lab, that tiny little skin contaminant is going to falsely signal a massive, life-threatening blood infection, which triggers hospital panic and a whole barrage of heavy, unnecessary antibiotics.
But if there is an undisputed king of the lab VIPs, it is cerebrospinal fluid, or CSF. This is the precious fluid, safely surrounding your brain and spinal cord. It's universally considered the absolute highest priority specimen in the entire hospital, because suspected meningitis can be immediately and rapidly fatal. When a CSF sample is drawn, someone physically rushes it straight to the lab for urgent cell counts and cultures. No delays, none. Lives literally hang in the balance of that specific sample's transport time.
We've also got anaerobic specimens. These are endlessly fascinating to me because they completely flip our everyday understanding of life. These microscopic organisms are incredibly vulnerable because they are actually damaged or even entirely killed by exposure to oxygen. For our anaerobic cargo, oxygen isn't life-giving, it's a deadly toxic threat. Because of this, they require highly specialized, perfectly air-free transport systems. Casually opening the tube to room air for even a second can completely destroy the very pathogen the lab is desperately trying to identify.
And finally, Part 5, reading the results.
So we've successfully collected the specimen, transported it, and protected it. Now, the clinical microbiologist has to play detective, and in biology, things are rarely black and white. Check out the stark medical difference between saliva and sputum. To the naked eye, they kind of look the same, but medically, worlds apart. Saliva is just packed with your normal oral flora, the everyday, totally harmless bugs living in your mouth. Sputum, though, is coughed up from deep down inside the lower respiratory tract. If a nurse specifically asks for sputum, but the patient just lazily spits saliva into the cup, that specimen is useless. It won't help diagnose a deep lung infection like pneumonia at all. The lab will only see the mouth bacteria, and they'll miss the real disease entirely.
And this perfectly highlights a massive, everyday challenge for the microbiologist, telling the difference between normal flora and a hostile pathogen. Listen, simply finding bacteria under a microscope does not automatically mean there is a disease. Human beings are literally covered in bacteria. The detective in the lab has to weigh a bunch of complex clues. What's the exact quantity of the bacteria? Is it a pure growth, or is it mixed in with a bunch of other stuff? And crucially, what was the exact anatomical site of collection? Because a bacteria that is perfectly normal and actually helpful in your gut becomes a deadly pathogen if it somehow sneaks into your bloodstream.
Which leads us right into this extremely important distinction that dictates how medicine is actually practiced: colonization versus infection. Colonization just means microorganisms are present on or in your body, but they aren't causing any disease whatsoever. They're just hanging out peacefully, paying rent. An infection? That means those exact same microorganisms are now actively invading your tissues, multiplying completely out of control, and producing a hostile host response. They aren't hanging out anymore, they are staging an all-out attack.
So, as we wrap up this explainer, I want to leave you with a rather wild thought. Where is the invisible line on your own body, right now, between harmless colonization and a hostile infection? At this exact second, you have millions of microscopic organisms living on your skin, in your mouth, and in your gut. They are perfectly peaceful until something in your immune system or environment shifts. And the moment that invisible line is crossed, one of those tiny little organisms might just be scooped up into a sterile plastic tube, ready to begin its own perilous hero's journey straight to the clinical microbiology lab. Keep learning, and I'll catch you on the next explainer.